Thank you very much, and thanks for coming on this beautiful Saturday morning. My name, as Anissa said, is Leslie Kenna, and I'm a clinical pharmacologist. And that means that I work on trying to figure out how to make drugs as safe and effective as they possibly can be. But today I'm going to talk about something a little different. I'm going to talk about a tool that scientists use to discover new medicines. And I'm going to work over here just so I can run my slides. And my talk is how fireflies light the way to new drugs. Now, one of the most important things in science is to acknowledge all the help you get. And that's where I want to start. Dr. Doug Ald at the National Human Genome Research Institute at the NIH, a terrific scientist and a great person, gave me the idea on how to run what you'll see as the Luciferase assay. And he helped me get the materials and walk me through the demonstration. So I am very grateful to him. The Promega Corporation provided some materials for the reaction, which was an extremely generous donation on their part. And I want to thank Patty Wooding and Anissa Ramirez from Yale University for the Science Saturday program, because they did a lot also to help me get here today. OK, so this is a picture of my 4-year-old son catching fireflies in the yard this past June. And here he is. And I don't know about you, but no matter how old I get, this just never ceases to be fun for me. I mean, if I wasn't taking the picture, you'd see I was out there running with him, trying to catch them. And then, I don't know if you could see here, just when he stopped trying to chase after them, there was one right there behind him. But fireflies are not just fun. They're also a very important tool. They've been an important tool for scientists to discover new medications. And so I don't know about when you saw the title of this talk, what you were imagining. But were you thinking that, do you think that scientists collect a lot of fireflies, stick them in a jar, and then go and look in the medicine cabinet? Well, that's one way. But what I'm going to talk about today is even more exciting than that and pretty sophisticated stuff. So to get to that point, we're going to have to talk about a couple other things. But by the end, I think you'll have a good understanding of what I mean and what scientists are doing. So the first thing we're going to talk about is, why do fireflies glow in the dark? From my perspective, it always seemed like I didn't really care that much because it just was fun. But they're doing something important when they're glowing in the dark. Second, how do they glow? What is it that makes fireflies glow? We'll talk about that. And then finally, we'll get to the big question, which is, how have scientists used this glowing to find new medications? And I want to point out that I'm not just going to be standing up here lecturing to you the whole time. I'm going to do some demonstrations and talk about the science so you understand it. And then after, when I'm done, you'll have plenty of time to play around with the things that I show in my talk. So we're going to learn a couple of big words today. And I thought, let's start out and get one out of the way right away. Here's one. Does anyone can somebody say this word? Bioluminescence. Have you heard this word before? OK. All right. So great. This is a very educated group. It just means living light. Fancy way of saying glow. OK. And what do you know about fireflies? When you look at them, is the whole firefly lighting up, or where is it coming from? Yeah, well, if you turn it over, they call that part here where the glow is coming from, they call that the lantern. OK? And so does anyone know why fireflies glow in the dark? Yes?
It's so that they can mate with other fireflies.
Yes. OK. I don't know if everybody heard that, but fireflies glow in the dark because they're searching for mates, and by flashing, that's communicating to other fireflies to, come find me. But fireflies are not the only creatures that are bioluminescent. There are a lot of other living things that bioluminesce. And what's interesting is that they all do it for very different reasons. And here's a list of reasons. 1 is finding mates. 2 is to lure prey, or find dinner. 3 is to attract some kind of insect or animal that's going to spread their spores around. And then 4 is to evade predators, or to avoid being eaten themselves. Okay, and I'm going to show you some images of these things in action. For example, though, to evade predators, there are some animals in the deep sea that they're able to release that bioluminescent glowing stuff, and they spread that around so that predators get confused when they're coming, and then they can't catch them. Okay, has anyone seen this before? Okay, what does this guy do? This anglerfish. Okay, right, so this guy's got a glowing thing on top of his head. This is a fish. He lives at 225 to 12,000 feet below the ocean surface. So does anyone know about what it's like down there? I heard someone say it. Yeah, very, very dark. You can't see your finger in front of you. Anybody have a guess what % of animals that live down there are able to bioluminesce? 10%? That's a good guess. It's higher. 20, even higher.
40.
But keep going. Higher. 90%. Is it 90% of the creatures that live at those deep depths in the ocean can bioluminesce? And so this is one creature. And I'm going to show you a video of him in action. Him or her, sorry. So what you're seeing here is the glow of the bulb on top of its head. And inside there are microorganisms that bioluminesce blue. And this thing is swimming around. And it's he's glowing. It is glowing. And this fish is looking for food. And so, I'm sorry, it's very dark, but that's how it is down there. And we couldn't see the glowing if it wasn't like this. Okay, I believe that's just some other kind of bioluminescent organism that's down there in the ocean with it. And I have to point out that I took a lot of these clips from YouTube because I don't have the ability to go down into the ocean and collect these myself. So there's another organism that's bioluminescing down there. Okay, so this is how he gets his name. The anglerfish, it's like angling with a fishing line, swinging it around. Oh, turn off the lights? And then here comes a cuttlefish, which is attracted to this light. And I believe it's because, down at that depth, you have to remember 90% of things are luminescent. So this cuttlefish is looking at it and thinking, well, maybe that's food. And so I think the anglerfish just ate him. Kind of cruel. Okay. So we've seen fireflies light up to mate, anglerfish light up to attract prey. There are some mushrooms, fungi, that bioluminesce, and you can see it at night. There's kind of a glow on the forest floor. And these are believed to be doing it to attract animals to spread their spores around. And does anybody know where this is taken? Yes? Yes. Have you been there? Yes. Maybe you should tell us about it. Okay. And so, so this is, they call it Bioluminescent Bay in Puerto Rico. It's this body of water where they have just an incredible concentration of these bioluminescent dinoflagellates. And dinoflagellates are simple organisms that have a little whip-like tail and they move around. But when they get disturbed, they glow. And if you run your hand through the water there, the water lights up. So that's what this person is doing. And apparently, the reason why they do it is they do it to avoid being eaten. And how it works is if they sense that something is going to if they get disturbed, if they're afraid that something's going to eat them, they light up, and apparently it attracts bigger fish to come into the area. And so then those bigger fish then eat the smaller fish that was about to eat them. And here is a little video of somebody throwing a rock into one of these bodies of water that bioluminesce. You'll see. Cool. I want to see that again. I never tire of looking at that. Hold on. I love that. Okay. So we've talked about why fireflies and other bioluminescent creatures glow. And now we could talk about how they glow. Okay, so I'm getting back to a picture I brought up earlier where we talked about how the fireflies glow seems to be coming from this lantern. Well, what's happening inside that area is that there's a chemical, luciferin, and a protein, luciferase. And when they come together in the presence of oxygen and ATP, does anyone know what ATP is? Energy. Yep, that's the cell's energy source. When all four of those things come together, the firefly gives off light, and it creates this other, just alters the protein. What's interesting is why fireflies glow. Can anyone guess why fireflies might glow? This is a tough question. I found it in doing my research for this presentation. Well, one scientist believe that it may be that, since you need all four of these components to get the lighting, to get the light, for the reaction to occur and to generate light, if one of these components is missing, the reaction stops for a little while. And it may be that in this area, the firefly runs out of oxygen temporarily, and so it has to build up oxygen again, and then it can glow again. So, and this is happening at a rhythmic, every 6 seconds, the typical firefly that you see on the northeast is glowing. And so I thought that was very interesting, that there's a very chemical reason, or at least a chemical mechanism for this flashing to occur. Another interesting point about the firefly, the light that comes from the firefly is 90% efficient. Now, could somebody explain to us what I mean by that when I say efficient? Okay, by efficient, I mean what percent of the energy that's created comes out as light versus heat. Now, when you touch a light bulb that's been on for a while, you usually shouldn't do that because it's very hot. And that tells you that light bulbs are not very efficient at converting energy into light because they're giving off a lot of heat. Does anyone want to guess what percent of the energy that goes into a light bulb comes out? 40. It's even lower than that. Yes. Great. Yeah, it's in the range of under 5% of the energy that goes into a light bulb comes out as light. Most of it is lost as heat. So fireflies are incredibly efficient. And that's something that's important in biology. Why do you think it would be a problem if the firefly generated a lot of heat when this reaction occurred inside of it? Yeah, it would burn you, and it would burn the firefly himself. Just like us, we have a very narrow range of temperature that we can be at. We're normally at 98.6, and if we go a couple degrees above that, it's a sign that we're very sick. So the same thing with fireflies. They have to keep it in the temperature range. So very interesting. And here's my really corny joke. I say that fireflies are the ultimate source of green energy. Ha, ha, ha. Really bad. OK, so now I'm going to get into a demo. And I'm going to ask for a volunteer. Did someone come volunteer? The first hand I saw was back there somewhere. Yeah, can you come on up? OK, can anyone tell me what this is? Does anyone recognize? Yes? A glow stick. OK. So here, you can come up too. Come help me. All right, so if they're glow sticks, why aren't they glowing? OK, so can you break them? Break it? OK, what did you hear? Okay. Does anyone know what happens inside of a glow stick? The first hand. Okay. Yeah, do you want to turn the lights up so we can see the glow sticks? Perfect. I don't need to be here. Everybody knows all this already. So the glow sticks have, like a firefly, they have a couple things inside that when mixed together, they give off energy, and that energy comes out in the form of light. You want to hold it up? Anyway, and when you hear the glass breaking, that's just the process of you're breaking it, some vial that's inside there, keeping those chemicals separate and getting them to mix together. Thank you. You can keep those. And we have plenty of glow sticks for everybody to play around with that afterwards during the time for hands-on. So, okay. Now, so the question I have for you now is, are fireflies just like glow sticks? What do you think?
No, because they don't have glass.
No, they don't have glass. That's true. Anything else? How are fireflies? Yes?
Fireflies can keep going on after a little while because they keep going off and back on. But if your glow sticks run out, they just stop.
Excellent point. Anything else? Back there? Uh-huh. Awesome points.
The color is only like yellow.
Oh, that's an interesting point, too, that the glow sticks come in a range of colors, but fireflies are only green.
Well, the glow sticks can't get oxygen like the fireflies do.
Okay. So that's interesting. So you get back to the point of why it might run out, because fireflies, they use oxygen, and you know the glow turns off in the firefly when the oxygen runs out, but then they can replenish that. But within a glow stick, it's closed, so once the whole reaction is over, when it runs out of the materials, the reaction's over. Anything else?
Can I just ask, do fireflies just glow at night and not during the day?
Do fireflies just glow at night and during the day? I don't know that. I believe so, but I would have to get back to you on that.
I asked because I know dinoflagellates,
Uh-huh.
they have a circadian rhythm, so they need to be exposed like 12 hours to daylight and then 12 hours of darkness. So
Okay, that's really interesting. And I guess, so she brings up the point. She asked if fireflies only glow at night. I think it's true. I'm not certain, though. But because I know, the reason why I've said I'm not certain is that glowworms, which are the pupa stage of fireflies, they glow underground. And so they have that ability to glow night or day, but because it's dark underground. I don't know if it's because of darkness, which triggers it. But this woman brings up the point that the dinoflagellates, which were those bioluminescent things we saw in the body of water that light up when you splash the water, they don't glow during the day. They only glow at night because they need to build up some energy from the sun. Interesting. Very interesting. One more point?
The fireflies are bioluminescent. Well, glow sticks are.
Excellent. Excellent. I feel like I must have paid him. You're going to think I paid him because I was just about to make that point. The big difference between fireflies and glow sticks is that fireflies are bioluminescent and glow sticks are chemiluminescent. And bioluminescence is, it's called bioluminescence because it's generated, one of the components that go into making the light is protein, and that comes from just a living organism. Whereas chemiluminescence is made all by chemicals, and that's something you can make in a lab. Yes? Yes, yes, exactly. Yeah, it's all chemicals. Good point. Very good point. And the other thing that's common between the two is that they're both, I think, efficient in terms of making light from the energy of the reaction, because it's not hot when you put a glow stick on your neck. You know, it's cool to the touch. Okay, so, well, that was all nice seeing what firefly luciferase is like, but I think you'd like to see the real thing, So I have a demonstration of Firefly Luciferase. After I mix it, we'll turn the lights down, but we're not going to do it right now. Actually, no, you can leave the lights on because I'm going to pass it around in a box. Okay, so this is going to take a minute or two. I am going to mix here. Scientists have isolated luciferin and luciferase, the protein and the chemical that go into the reaction to making a firefly glow. And what I have here in this tube is luciferase. This is the protein. And in this tube, I have luciferin. And I'm going to mix the two together, and we're going to get glow. So on their own, I mean, you could turn off the light if you want to show people that on their own they're not glowing. Can you turn off the light for a sec? It's just going to be dark, and you're going to see nothing because, oh, it dims. Thank you. Yeah, on their own they're not glowing, right? Do you agree? Okay, so turn it back up. And now, actually, let me take just a teeny bit more of that. Okay, so I'm going to take about a milliliter of this luciferin. Put that in there. I'm going to take a microliter of luciferase. Oops. Try that again. Put that in there. Okay. And let me mix it up. Yeah, lights up. I don't know if you're going to be able to see it. I have a little box to put it in. You can see it. It might be hard to see from so far away in the room. I'll mix up another one or two. I'm going to put it in this little box, and we can pass this box around. I'll get a couple of them going. And you can see. So as you can see, there was no firefly. It was just the chemicals from the firefly. Yeah, OK. So when it's in the box, you can see it. I'll make one more, and then we'll pass them around the room. Very cool. Thank you. What's that? It adds oxygen to it. It makes oxyluciferase. Yes, because that's not a reversible reaction. Okay, here's one more. And again, it'll go around, and then also later on during hands-on, you can come play with this a little bit too. There's another one. Yep, take a look. Tell me if it's glowing.
That's cool.
Yeah. Actually, I can make one more. If you don't mind me taking another minute just to make one more. Then we can get them. What was that? How many amino acids in luciferase? I don't know offhand. I'm sorry. Yes? Thank you. That's the introduction to where we're going to go right now. When I go back over there. All right. There we go. All right. Okay, so somebody just asked this very question. So how did I get this luciferase? Well, one option would be I could go out and collect a lot of fireflies and get the chemical from inside them. There's a couple problems with this. Does anyone want to point out? Yeah? You'd have to catch a lot of fireflies. Okay. Did anyone else want to talk about why this might not be a good thing to do?
Fireflies might have a limited amount
Mm-hmm. Okay, yes, it'd be hard to come by. How about, okay, someone back there? Yeah, you'd have to kill them. It wouldn't be very nice to the firefly or fireflies.
You still have to separate it and do all these other processes after you get it, right?
Yeah, and that's the other problem, is that actually you might not, I don't know if you could separate it, because someone asked before, is this a reversible reaction? Does the oxygen bind and then come off again? And I don't think so. So it's not really a good way to do it. And then the other thing is that, what was I thinking? No, I think that's the whole point I wanted to make about that. Not very nice to fireflies. Not a very, oh, I'm sorry. It's October right now. You know, you'd only be able to do this demonstration 1 month out of the year. So we need another way. And there is another option. We can have cells make it. Because what do you think we need to have cells make it? I think, have you guys?
The 4 ingredients that were on the page, like oxygen, luciferase, and
Luciferin, yep. He's got it. He said we would need the 4 ingredients. So how would we get those 4 ingredients? One of them is a protein. Is that hard to get?
no.
How do you get the protein? Yes, excellent. So he's talking about genetic engineering. Okay, so, and this is the big difference between chemiluminescence and bioluminescence. If it was just 2 chemicals coming together to make the glow, you could easily just make that in a factory. This, since one of them is a protein, it involves some kind of cell or cell system to create the luciferase. And what you need to do that is either the RNA or the gene for this luciferase. And that's been isolated. So I'm just going to go through this on how you can get cells to make luciferase. Well, as long as you have the luciferase gene, and a gene is just an instruction telling a cell how to make something, and if you insert that gene into a cell and then trigger gene expression, then those cells will start making luciferase, and then you can collect it and put it in a little tube like I just showed you. And the process of putting genes into a cell is called transfection. All right, so we've talked about why fireflies glow. We've talked about how fireflies glow. Now the final big question is, how do scientists use bioluminescence to find new drugs? Okay, pop quiz. Why do we take medicine? Okay, yes. So we take medicine when we're sick, right, to treat us. It may cure, it may make us feel better, depending on what the disease is, what the drug is. Now another question is, why do we even get sick in the first place? Bacteria, okay. What do bacteria do? Okay. And what does that virus do? Yeah, it gets into your body. Do you know it specifically goes inside a cell, right? Okay, and a virus goes into a cell, and it takes over the cell, and it makes the cell do something the cell doesn't normally do. Did you want to add something? Okay, yeah, some things can mutate the cell. It can get into the DNA and cause it to do different things. So the basic common thing going through this is we get sick because certain cells in our body stop working properly. And it might be because of some infection, or it might be because of something else going on inside the body causes cells to stop working properly. So one of the reasons scientists study cells is to understand why cells stop working properly or why they get sick. And to test new medicines for treating these sick cells. So you'll look at a cell that's not functioning and compare it to a cell that is functioning, and by seeing what's different, you understand the cause of the illness. And then likewise, if you then treat it with a drug and then look, you could see if that drug is working or not. So, and I know that's very basic, but I wanted to bring that up as a way to introduce why scientists use luciferase to study cells, or how they do it. Well, what they do is they identify a part of the cell that they want to study. Have you guys taken biology, cell biology? Do you know parts of the cell? Can somebody name some parts of the cell? I heard mitochondria, cell wall, lysosome, cytoplasm, nucleus, ribosome, yep, ER, endoplasmic reticulum, that's hard to say. Excellent. So you're familiar with cells. You've talked about cells a lot. Now, the things we're talking about, the things you mentioned, were things that you can stain. If you've seen cells assays, and I'll show you that in a minute. Sorry. Biologists add coloring to cells to light up these different parts of the cell. Well, luciferase is exciting because we have the gene, and it can be put into the DNA in a cell just about anywhere. So it means you can light up just about anything, and it doesn't have to just be the endoplasmic reticulum. You can light up any kind of protein that's made, because the cells are doing all kinds of things. You know, they're getting infected by viruses. They're, you know, they have all these different receptors on the surface that are binding with different drugs. And those are the kinds of things that in normal, when I was growing up at least, when I was looking at biology textbooks, you couldn't see a picture of that. You couldn't see a picture of those receptors because nobody had a stain that could go and stain those things. But now we have luciferase genes that you can, a luciferase gene that you could put into the DNA in a cell right next to the genes for those other things. And you know what the luciferase does is it tells the cell to put a little tag of this little green protein, connect this little glowing green protein to this other protein so that when the cell starts making that other protein, it'll now light up. And I'm just going to show you a little cartoon of what I'm trying to say. So imagine that you're looking at some receptor in the cell for something. I don't know, let's say it's a smell receptor in the nose. Okay, this is a receptor that when you smell donuts, this receptor gets excited and it does something, and that's why you smell donuts. Well, we don't have stains to look at that. And so this is, without luciferase, I'm just showing you an outline of it. You really wouldn't be able to see that. But with something like luciferase, if you tagged the gene for that particular receptor, now once you did something to trigger the gene expression, it would light up green. So this is really exciting. And this is just an example to show you what luciferase has been used to do. This is a plant, and the luciferase gene was put into protoplast cells. And this plant was, they put luciferin, the chemical that reacts with luciferase, in the water that they used to water this plant. And so then the water was drawn up into the plant. And then when they turned out the light, they saw that it glowed. And this plant normally doesn't glow in the dark like this. I saw something on Wikipedia, if you look up bioluminescence on Wikipedia, they have a wealth of information. And one of the things they have is ideas that people have proposed for uses for luciferase. And one of them was to have luciferase put into trees on the side of highways so that they wouldn't have to have light bulbs. And it would reduce energy costs for the state. Can you imagine? Okay, so this is getting back to what I was talking about earlier, about the difference between luciferase being used to look at things in a cell versus using stains. Now, I'm bringing this back to cell division because I wanted to bring it back to the idea that we're going to be looking at cells to come up with new medicines, to develop new medicines. And cell division I just put up there because I thought everybody has studied cell division to some extent. So does anyone know the different stages of cell division? Yes? Excellent. Excellent. So these are different stages of where, you know, when one cell is going to divide into two. This is how these are different stages by which the cell divides up the cellular components and then breaks off into two cells. And when I was in school, we learned about it in this way, and we looked at stains. Nowadays, with bioluminescent proteins, here's a whole new way to see cell division. Watch this. And this, okay, I'm going to show you that again now that the lights are off. Whoops, sorry. Just one more time. These are pig kidney cells going through mitosis. And it's somebody tagged the tubulin in the cells. Tubulin is a structural component of cells, and it comes to life during cell division because tubulin is needed to move the different components of the cells into two different places. So it lights up a lot during cell division because it's very active during that time. Anyway, so I thought that was very interesting. I do have to point out, however, that this is a different protein than luciferase. Can anyone guess what protein was used in this particular study? GFP. I heard GFP. What does GFP stand for? Green fluorescent protein. Okay. So green fluorescent protein, and you're probably thinking, wait, we were just talking about luciferase. What's this about green fluorescent protein? Luciferase and green fluorescent protein are both bioluminescent proteins that come from living things, of course, if they're bioluminescent. Green fluorescent protein comes from a jellyfish, though. In 2008, the Nobel Prize in Chemistry was awarded to 3 people for the discovery of this. Oh, thank you. And because it's been incredibly useful. Now I'm going to explain to you how green fluorescent protein works and why it's different than luciferase. But I thought you might want to see a little picture first. So jellyfish are bioluminescent. This particular jellyfish is not, I don't know if it's glowing green or not. I don't know if it glows white or if it's just because of the quality of the video that it really is green, but it looks white. Anyhow, there are green glowing jellyfish out there. But they glow, they bioluminesce only when light is shown on them. This particular video is with a black light. But with green fluorescent protein, these proteins are fluorescing, and that means that they're taking in light of a particular wavelength. In the case of GFP, it's blue light, and then they reflect it out green. Now, I'm just wondering if anybody, does this sound familiar to anybody? What is that? Someone's raising their hand? Did you want to add something? Okay, so hands. If anyone wants to, okay. All right, so these are glow-in-the-dark stars. And just like we did with the light sticks, I want to talk to you about how glow-in-the-dark stars are similar to GFP and how they're different. Well, they're similar. How do you get a glow-in-the-dark star to glow? Yes? Yes. Okay, so you have to let it sit in the light for a while, and then when you turn out the lights, they glow. Can you tell me something about when they do glow once the lights go out? Does it glow for just, does it flash, or what does it do? Okay, so if you go to bed and you have the glow-in-the-dark stars on your ceiling, what happens when you wake up in the morning? They're not glowing anymore. But they glow, you know, for a little while or throughout the night, and at some point they burn off their energy. Well, my question is green fluorescent protein like a glow-in-the-dark star? Well, it's similar in the sense that you need to shine light on it to cause it to glow. However, glow-in-the-dark stars are actually phosphorescent. And the difference is that they emit this light over a period of time. They don't just throw it right back all at once. It's a slow release of the energy. So just to review, we talked about why fireflies glow in the dark, how they glow in the dark, and we talked about how scientists have used bioluminescent materials to find new drugs. And along the way, we learned some pretty big words. We learned how bioluminescence is similar, but not exactly the same as chemiluminescence. And we learned how fluorescence is likewise similar, but not the same as phosphorescence. So now, I don't expect you to have gotten all that. I'm certainly not a person who gets everything by just listening to someone talk about it. We have some hands-on activities. You can come and take a look at these luciferase reactions that are going on inside the box. You can play with light sticks. We have glow-in-the-dark dinosaurs in the box there. You can play with flashlights and then close it and look. We have some glow-in-the-dark crayons, and then I have some more videos of other bioluminescent creatures if you want to look at that. So thank you.